Well, thanks for having me, and I know I'm the last speaker here, and drinks are awaiting you. I am tempted, as the previous speaker said, to comment only on what he talked about, because it is the case that kind of talking straightforwardly about failure, and I'll put that in parentheses, is an important part of learning how to succeed. I would only say that the evolutionary the evolution analogy is apt, and evolution can only be thought of as an ensemble process. It's cruel at the individual organism level. Investors figured that out in this field a long time ago by creating portfolios. And what I'm going to tell you about is a 25-year journey of a firm that I started largely to counteract that. That is the unfairness, in my view, that entrepreneurs have to put all their eggs in one basket, because that's how they're measured by investors. Investors get to put all their eggs in every other basket. And while they don't get as much as the upside of a single winning lottery ticket, on average, if they are good and get better, they win. So I'll leave that with you at Q&A. I'm happy to come back. But I think that among the ways to offset this is to think the way organisms think, which is that at the population level, there's learning that goes on, even if some subset of them lose in particular competitions. So with that, I'm going to change a bit the topic and tell you a little bit about this topic. So obviously, I'm in Cambridge, one of the bastions of world-class biology historically and today and well into the future. And I'm starting here just to kind of look at this from the point of view of the present and say, when nature and human intelligence collide, we kind of call that biology, we call that scientific discovery. And more recently, we've been fascinated by humans actually colliding with something that they created, which is computation, getting more and more into a thinking machine or a form of intelligence. And so we call that artificial intelligence. And the reason I bring this up in the beginning is I want to try to define for you a concept that might help us reconsider what it is that we do in biotechnology. And so if you think about the human-computer interface, it's a whole new science, because humans are struggling with understanding what the computers they've created do and how they do it, and it offends them greatly. They'd like to think that computers are dumb, just like they thought most of nature is dumb, and that they're the only smart things alive. But in reality, we're beginning to realize that the product of our great ingenuity has produced things that kind of seem like they're thinking in some ways. We can argue whether it's reasoning, whether we can argue whether it's sentient, and we'll do that in Q&A. But the point is, there's a new form of intelligence in town. It's called artificial intelligence. And kind of buoyed by that, I want to present to you, or represent to you, how we should all think about what it is we work on, which is fundamentally a form of intelligence, which is simply nature's intelligence. Nature, in every which way, is intelligent. It involves designs that adapt, that dictate behavior. It has every bit as much the ability to learn. It simply encodes it in the form of DNA. All sorts of DNA markings, all the downstream effects of those, but ultimately DNA. And but for the fact that our neurons encode our learning in a different system, just like the silicon that computers use encoded in yet a different system, we somehow, as humans, have deemed nature not quite intelligent, but we're willing to say that these silicon machines are intelligent. So I would argue that we're entering kind of a reconsideration of what it is to do biology, and that is one form of intelligence, human, trying to cope and understand another form of intelligence. And this is going to sound very presumptuous of me, so bear with me. I'm simply the messenger here of some errant thoughts. But I would argue that we're entering an era where humans now have two forms of intelligence to deal with. One is artificial and the other is nature. And if you look for signs of intelligence in nature, you can't but find them everywhere. A virus that just about took down the entire human population and kept matching, wits with a set of immune systems, how is that not intelligent? If this was a computer virus, we would be thinking it's artificial intelligence. But it's a natural virus. And cancers that can basically resist wave after wave of attack. So I think you might think it's just semantics, what I'm calling it and what you call it. But the reality is, if you think of it as intelligence, I suspect we'll be approaching some of these problems differently. And the reason I say that, one last point on this, is the following. I think that the way we've been doing biology, which has humans as the intermediary, can only work on a small subset of biological problems. It's the ones that lend themselves to simplification. We call that abstraction as humans, big word. But simplification, which is that we can cope with systems that with a few pieces interacting in a few ways can make for a review paper. But if you had systems, as we know we have in our cells, which have thousands of parts, and I'm simplifying, maybe tens of thousands, hundreds of thousands, interacting in thousands of ways to define a particular state, and that state change can best be understood by using all of those components in a model to explain what's happening, well, humans, I am afraid, at least today, are out of that game. But the computers we've created, extensions of our human brains, actually can cope with that complexity. So what happens if models that have billions of parameters, or however many we need, that we now have the ability to create, actually begin to emulate and better represent what we're observing in nature, even though the humans, the experts, can't explain it and can't understand it? Are we really willing to say, no, we don't want to work on that. We're not going to allow that to be published. We're not going to use that to discover or develop drugs. That's what we're facing, in my view, right now. And it really is, and I'm sorry it's the end of the day so I can speak conceitfully, but it is the conceit of humans that is going to keep us from being able to go after a vast number of new biological frontiers. And I am, at 38 years into my career, dramatically more optimistic about what's going to be possible and is being done today than I have been in the past, including when the genome was sequenced, something I was involved with, including all the different breakthroughs. What's ahead, those of you who are students today, is quite literally a gain-of-function kind of inflection point, in my view. So with that as an opening, and if anybody's interested in this stuff, there's not that much written on it, but we wrote a piece earlier in the year called Polyintelligence. This was just to lay out the basic principles. And I'll just say one last thing. The actual point of the letter was to draw inspiration from Leonardo da Vinci, for whom, and he must have had great instinct, this notion of nature, clearly if you read his writings or what's been written about him, you kind of see that he absolutely revered nature as a form of intelligence. He did not view it as a static thing. He did not view it as something that was subject to human control. In fact, if anything, he viewed it as a much greater thing. And all I'm telling you is that today, so many hundred years later, we've invented machines by which we can actually treat it and learn from it as a form of intelligence. So the rest of my talk is not going to have a lot to do with it, except subtly. So let me explain what I mean. What we do as Flagship Pioneering, the firm that many people in the room actually have come over to talk about, and we're very excited to be working more and more with colleagues in the UK and in Cambridge in particular, is really to create new platforms. And when we think about platforms, bioplatforms in our work, we very much are trying to understand and deploy technologies to be able to do a better job at therapeutics, prevention, diagnostics, but all of them at their core is an attempt to try to harness this intelligence that we are either up against or want to emulate. So I'll say a little bit about Flagship so you get a bit of a sense. We're kind of an odd entity in many ways because we're not a single entrepreneurial startup. We're 45 of them. They've all been started the same way. Moderna was our 18th. It was numbered 18th. It started in our labs. The first 6 patents that govern Moderna's IP is entirely based on the work that was done in our labs. A lot of the academic work informed what we could do, but in fact, a lot of the work that preceded us had been stuck, completely stuck, in that one couldn't overcome the immune response of these RNAs that were being used. And some of the scientists that deserve a lot of the credit came up with ways to overcome that. But more broadly, what we do is conceive, create, develop, and launch companies. So we are a company of companies. And we specialize in first-of-their-kind platforms. In other words, we don't work on things that are extensions trying to compete. We just simply try to figure out what should exist 5 years from now, and can we bring it to bear today? You could imagine just how much failure is involved in that activity. But that's what we try to do. We do this in an institutional way. I mentioned earlier that for the first 13 years of my career, I did things as an entrepreneur is supposed to do, one at a time. And after a while, I thought, well, why can't you do it multi at a time and just get large teams of people to collaborate and share risk and reward? That's what we do. So we have an institutional way of doing this. And what's really interesting, and I'm not going to have a lot of time to talk about, is what's resulted is actually an ecosystem of companies that are part of one family. Just like if you're in one family, you have one large culture that kind of governs how you behave, even though you might be an individual with your own distinct beliefs and character, etc. our companies come from the same place, they have the same source of IP, and therefore much is shared. All the capital access and people access and relationships are all available to all of the companies. So that's another interesting experiment. I'm going to skip this just to say at the core Flagship is about 1,000-person team. 500 of them are core members of the actual operating entity. Another 500 are in entities that are within a 3-year cycle of being spun out. And so all of them operate within, and then every year a subset of these companies end up becoming spun out and develop on their own. I'm going to, because of time, skip ahead. If any of you are interested, you can look this up. We say quite a bit about this on our website. We have a stage gate way of thinking about this. We don't look for brilliant ideas. We don't come up with brilliant ideas. We try really hard. So what we do is about 100 times a year, we conduct explorations that lead to some things that after some iterative cycles, people can't tell us why it won't work, except it's never been tried. We go into the lab in the next phase and try to reduce them to practice. In a short amount of time, with a limited amount of money, those are extremely important evolutionary constraints. In fact, I could tell you right out of the gate the thing that I used to hate to hear as an entrepreneur, and I hate to say now, which is that many companies fail because they get access to too much money. Many companies also fail because they get access to too little money. That's also true. But those are different phenomena. And if you take the evolutionary selection pressure, there's no one thing you can know for sure is you could spend money. The question is what sequence and what do you spend it on? And how can you create some version of a Bayesian logic to inform you? You won't do that if you have enough money to do everything. So that trade-off is done for us in the second phase. The third phase is where we go in and build a platform. So if something fails to die, it deserves advancing. So one of the interesting kind of semantic issues I would say is that if we simply consider winning as nothing more than failing to lose, then if you just keep kind of getting chances at bats, is what we call in the baseball analogy, then winning comes from that. So we view it that way. If things in that protocol phase fail to die, we advance them, we create these new codes, and eventually growth codes. And in that model, this is kind of what exists today. These are these things emanating from the center. You'll see a bunch of numbered entities. One of the things we learned 25 years ago in the journey, which helped us, is that you don't want to name, ill-formed ideas at their earliest stage, simply because when they fail, then you have to go tell somebody that you no longer are working on Phoenix Rising or whatever name you give it. But if you call it FL118, it turns out nobody really loses a lot of sleep over moving on to FL125, which is exactly what our team does. As crazy as it might sound, you really do want to make it clear to people that this initial phase is very much this formative, iterative phase, and if it doesn't come out, great. There's an opportunity cost on working on the wrong thing. And so instead, work on something different. So that's the culture we've tried to set up. And if we add now the ones that kind of preceded that previous look and emanating from the middle, now the growth companies and the larger ones on the outside, this is what the picture looks like. I show this because I want to just flip it a little bit to come back to the polyintelligence and platform thing, spend a little bit of time on technology and science. This is the ecosystem that exists roughly today. I've skipped about a dozen little things, less than 10 people. But if these are all entities that are within their third to, let's say, 10th year of existence, and what you'll find is a whole bunch of them are in the novel biology target ID space. This is a very big unmet need in the pharma industry, no matter what they say. I'm sure there's tons of pharma people here, but people generally want to think we have all the biology we need. What we really need is better ways to drug things. Actually, we need a lot of everything. And so we have a number of interesting things in that space. But then, regardless of molecule class, regardless of disease, there's tons of unmet needs and breakthroughs that are awaiting to be happen. So this is just to show you a frame within which at least... Now, by the way, if you sit there and say, are you trying to kind of match how many small molecule, large molecule? Not at all. All this is emergent. It's completely emergent. Our scientists, about a couple hundred of them, who get to do this type of exploratory kind of future backwards research on what should exist, get to work on any number of questions. Whatever fails to die advances, and it so happens that they occupy a number of different areas shown here. Out of these platforms, though, if you don't get this picture, which is a set of products that are actually affecting human lives, hopefully, then you're not going to get the capital needed to actually do this upfront stuff. And so this is kind of an ecosystem view of the programs in preclinical, clinical development, simply to say that there's a lot of disease diversity. One of the advantages of not being a company with large sales today, we don't have any at Flagship, but individual companies, some of them have some sales, Moderna included, is that you don't have to keep doing more of that. So we have no incumbency. One of the things that you learn as an entrepreneur is the difference between an incumbency and an insurgency. So insurgencies are good in that they free you up to do totally new things. They're bad in that you don't have enough resource, nobody believes you, on and on and on. So this is kind of an insurgent portfolio of a number of therapeutic approaches or prevention approaches. And what's even more interesting, and I just show this largely to just zoom out, to say that these platforms, if you innovate in the platform level, have applicability far, far beyond the couple of hundred things that you saw. This is just a kind of recent cut. Every 6 months or so, our team goes and says, okay, what products, forget platforms, should exist emanating from the capabilities we now believe we have enabled through these platforms? And so how you convert platform potential to products and ultimately human impact, that's the journey. That's really the hard part. But by the way, one of the things we've learned doing this at a fairly industrial scale, hundreds of times, kind of a year, every 2 years, is that the real value creation actually happens in reverse. In other words, if you can figure out what will have impact on lives, what NHS will pay for, which is tough to figure out, but for pharma, and work backwards, then the enabling technology you need looks very different than if you think of it from the platform point of view forward. That's one of the things that wasn't obvious to me when we went down this path. I give the analogy, maybe it'll stick in your mind, of people who must have at some point invented musical instruments. I have done no such thing, and I don't know if anybody in the room has tried to invent a new musical instrument. It's kind of a lost art. But if you did, now what would that feel like? Well, you would make something, put some strings or holes in some whatever tube, and ultimately you'd make sound. And that sound presumably would be different, otherwise it wouldn't be a new instrument. But once you hear that sound, the question becomes, what music do you play? And who's composing for that instrument? And the answer is not the one making the box or the instrument. And so how does somebody who's imagining and making a new musical instrument pair up with music composers and players for an instrument that didn't exist? That's the fundamental, gigantic problem with innovating with platforms in our industry. That's very different than the tech world, where platforms are largely a chassis on top of which other people make products. So it's other people's products that sit on a iPhone platform or a cloud platform or whatever. So we think a lot about these things. Let me get back to my original talk. But just these are things that as an entrepreneur you kind of want to think about carefully. I'm going to skip ahead just to say that there's every way you look. If you think from an intelligence standpoint, you'll find problems that recast themselves if you think of them as learning environments. So for example, when we did the human genome sequence 25 years ago collectively, we kind of were thinking, whose genome are we really sequencing? As though there is a genome of a human, and that's what matters. Germline kind of genomes were the thing. We all wondered back when, and at the time Solexa existed, it's a great company. We started a single molecule sequencing company called Helicos, which ultimately failed. It succeeded technologically. It developed $1 million machine in 2008 when the economy completely froze and you couldn't sell a $100,000 machine. And so that company perished, but the technology was very instructive. And one of the things it was instructive of is it allowed people to look at the single cell level and see how much genetic variation there is between somatic cells. So how much somatic variation. And when we glimpsed at that 10 years ago, we could see massive variation. We would want to ascribe that to error of sequencing, except we weren't amplifying. We were doing nothing. We were directly reading DNA. Well, guess what? Many, many years later, using Sanger sequencing and lots of different advances, we now, in a company called Quotient, which is actually partly based in Cambridge, the two Cambridges, is actually benefiting from the realization that there's a massive amount of somatic variation in our cells throughout our body, and that variation is, of course, selected upon. It would be hard not to be. And that selection that goes on in our bodies is not just relevant for T cells and B cells and cancer cells, but for many kinds of cells. And they contain massive amount of information that can inform target selection and how diseases are either resisted or accelerated. All things that, if you think about it as learning algorithms with lots of data instructing on what you can do, take a different form. So quotient, in my view, is very much an assumption that the nature's intelligence as manifest in intracellular or intercellular variation within a single person might be useful for drug discovery. Another project we have, Profound, has for the many, many years gone after understanding what are all these mass spec sequences that you could see that don't correlate with the coding genome. Many, many years ago, you've now read in papers that's coming out in the academic world. 5 years ago, we had already documented thousands and thousands of ORFs, human ORFs that do not correspond to what's in the protein databases, and that clearly we could show by RiboSeq, as well as mass spec corresponding sequences, are being translated. Many, if not most of which, come from known coding regions. These are not alternatively spliced things. And many, many of which we've correlated to certain changes that happen in disease, missing explanations for things we thought we had the right mechanism for, except there was a continent missing, and so on. I view that as another form of, if you think about how cells are operating at the level of complexity far beyond what we've forced it to be simplified to, then you can begin to see things. So if you believe things don't exist, you stop looking for them. This is like the old days when junk DNA was the in thing. This is 30 years ago when the sequence was being done. Well, guess what? Non-coding DNA is a lot less non-coding than we all thought. And that's going to become even more the case the more we look. At least that's what we're finding. A lot of modalities. Let me skip ahead. We've done quite a bit of work in gene writing. That is kind of not editing, but using the intelligence that's become kind of part of how human cells are being modified through mobile genetic elements. The single most kind of prolific gene class in the genome, mobile genetic elements, is not what we use or historically use to do gene editing. We're doing that now in this approach. But let me kind of skip ahead because of time. There's some covalent chemistry things that also lend themselves to thinking about nature in a very different way, but I'm going to skip all this. Let me just kind of get to the end, which is where I started, which is a little bit more about the role that AI is playing in fueling what's going on in the science. So Generate's a company that we started about 8 years ago that was, at least to our knowledge back then, the first to try to actually come up with learning algorithms to do generation of new proteins. Initially, we wanted to look at enzymes and lots of different proteins. Eventually, we realized that the single biggest value proposition is if you could basically computationally design or specify an antibody that could basically bind any part of any protein you wanted. That was the goal. And where we are on that journey is kind of quite a long way down the path, because if you integrate, which is what Generate did, the model work that went through generation after generation. We started out with transformer models, we went to diffusion models, we've gone to now some hybrids of LLMs and all sorts of versions of now multimodal and other models. The model part is going to continue to evolve. The experimental part was not actually industrialized back in the day because nobody made tens of thousands of antibodies. Worse yet, cryo-EM'd hundreds and thousands of them, which is the aspiration. We're doing hundreds now. And the learning cycle you get at looking at that variety of design, test, and model and learn cycles has been dramatic. Generate is just entering a phase 3 trial of an antibody that came out of this work a couple, 3 years ago. We have a dozen or so clinical programs that are in the preclinical phase, advancing to clinical, but where the initial proof of concepts are coming in through non-human primate work. We have done 2 human trials. Both have shown the antibodies computer-generated, combined where we want. And so that actually is, how biology is going to be done. It used to be that we used biological high-throughput techniques, directed evolution, libraries of different kinds, phages and yeast and stuff. But increasingly, we will use computational libraries, and the data that is basically helping them get trained are, to me, the transmittal of nature's intelligence to forms of computer intelligence, machine intelligence. And as one starts revving up with more and more capability, the other, I think, will reveal more of its complexity, not bothering to wait for humans to understand how all this works. And I know I'm saying provocative things. I'm hoping it will provoke you challenge that assumption. And maybe the last example that I'll give is a company that was recently revealed, although it's about 3 years old, a company called Lila. And Lila has been our internal effort at seeing if we could actually do the kind of equivalent of Waymo for science, for biological science. That is a self-driving, autonomous way to generate hypotheses in an arbitrary subsection of a field. Biology, we're also doing material science. To generate hypotheses, not one at a time, but hundreds at a time. To down-select from them ones that are prone to experimentation, to then, through agent-based systems, specify experimental protocols without human involvement, trained by all the experiments that have been described before in the literature and otherwise, run those experiments, that's the part we've had for a while, automated experimentation, and then interpret the data. And the question is, can you close that loop? And that's very much a work in progress. Over the last few years, we've shown closing of that loop in a few specific areas. And one of the things you get when you close that loop is you can run it over and over and over. And the problem, of course, is that you can't explain after a while why it's designing RNA in a particular way, LMPs in a particular way. This is very disarming for humans, as I said, because the question is, what's our role? So people are there now kind of thinking about what's humans' role, but the reality is, if you actually make these physical systems do this, it becomes even more interesting because you can neither understand why the hypothesis is the hypothesis, or why, because it's not sitting there explaining why it hypothesized what it hypothesized. Like in humans, if you hypothesize something, you have to explain it to your colleagues, to other people in the field, except if you're a physicist or a mathematician. There, you can levitate at your heart's content. I have a brother who's a theoretical physicist, and I always envied him because he got to make whatever size leap he wanted. We in biology have been gravitationally pulled down to entirely defensible, reasonable hypotheses. Just think about it. These things will allow us to at least defy gravity a little. I'm not saying it's going to get rid of gravity, but it's going to defy it a little, because we're going to get to do with the same cost, I believe, eventually when it's perfected, at the same cost, a hell of a lot more parallel experiments to reveal which hypothesis it was worth working on with the same amount of effort. That's going to be the interesting change. And so this is laying out kind of how the team at Lila is thinking about this. Just to say that you need to be very, very competitive at the reasoning thing. It's very hard to be competitive with people who are spending billions and hundreds of billions of dollars building reasoning models based on social media and reading all the scientific papers. In our hands, the fit-for-task versions of these models can, at least today, outperform what we're seeing out there in the more broad ChatGPT, the higher-end models for O and Claude, et cetera. These are different ways in which our team, and it keeps changing on a weekly basis, are trying to do the competition. The hash lines are human performance in similar tasks, and we're trying to figure out what is the level we need to get to using these models that allow us to really kind of let loose some of this capability. And again, when I say let loose, I don't mean the output of it. I mean the process of how you come up with a discovery so that we could then go in the lab and test for its usefulness. To do this, you also need to reimagine what a lab looks like. This looks like an otherwise ordinary lab, except the guts of this lab is where you're not, these machines are not made for humans. These machines are meant to interoperate. So if I put a skin around this, ideally someday, it would just be a black box. But internally, at least per molecule class, chemistry, covalent chemistry, RNA, cells, you can kind of come up with workflows that allow you to go through these at a very rapid pace. So early on in this process, I'm not saying that we've kind of reached the other continent, but this is something that we are very much encouraged by the early results and trying to bring about. We will fail hundreds of times more than we will succeed in this, but as did science on the way to becoming what it is we call science today, the prize is worth it. That's kind of the bottom line. Okay. I'm going to spare you this, which kind of says how all this in a fine-tuned way, all these different approaches and companies we have, starts redefining the entire ideation all the way to drug development and administration cycle. Maybe this is one thing worth ending on, since I'm timing it to 30 seconds now, and then we can have questions. This is remember the slide at the beginning where I said we've got these 36 companies that were in different areas? We've taken all of those logos and converted them into actually what they do. So just to give you a sense, and bin them by years, so every 5-year increment. And one of the interesting things is that against this if you look today, which we did about a month ago, at where AI is playing a complete enabling role or a large enabling role, you could see that in the last 5 years, it's absolutely everything that is fundamentally built upon these capabilities. And even going backwards, because these are previous companies are adopting these things, it's beginning to kind of penetrate heavily. And to be competitive, that's kind of what we think you have to do. So we view our world as kind of a periodic table of platforms and capabilities. It's not about the elements, it's about the compounds that you can make out of it. And my sense is that thinking about these compounds themselves, thinking about nature as a form of intelligence, will make this a far more fascinating journey, provided we're willing to share the steering wheel with what's around us. Machine intelligence, nature's intelligence. Thank you.
Fabulous and delightfully provocative in terms of what we can do and what the future is. So, any questions out there for Noubar? If not, I'll start for one. So one of the challenges that you've got, just sort of standing here from the pharma perspective, is the sense of the pace of the change is accelerating, no question. So changing the current physical infrastructure that we've got in the labs into something that is at that level of automation, beyond adopting the AI, that's a challenge. Have you found when you've taken some of the insights that you're getting from organizations like Lila that it's easy to transpose those into some of those earlier companies that you've got that were founded more than 5 years ago?
So I would say we're not quite at the stage where we are kind of bringing what we're experiencing at Lila into the other companies, but it's been a benefit that they've all been trying to do parts of this on their own, because since they're not trying to solve the generic problems, they're trying to solve their own particular problems. I'd say they're more familiar with how hard it is, and if somebody actually then comes and creates a standard way of doing this, they might be more open-minded to it than if people had never tried it. So it's almost like if you buy the wrong computer or the wrong, SAP system or whatever it is that and then all of a sudden you say, well, I don't want to deal with this, how do I do this differently? It might actually mean that you have to go through some pain before the gain of doing it differently. So I don't know how it's going to play out, but if you look at, I remember the first instruments I used in the labs when I was a graduate student had no computers involved with them, and they at most had little controllers. Then the kind of mini computer, and I used to be in the instrument business 30 years ago, mini computer, personal computer, and all sorts of limbs and stuff. That transition was similar, right? People used to do science without it, and then you kind of tried to do it yourself. So I'd say today there's a huge business of people consulting to help everybody do this themselves. And we'll see if that's going to be feasible. I think what's going to take is people who say, you know what, I'll solve this in a general way and then customize it for people. And so we'll see how that journey goes.
Okay. There's a question at the back there. Thank you.
Questions or criticisms, that's okay too. Oh.
Hi, thanks so much. Very thought-provoking. I had a thought about your failures and what you do with them. Just thinking, if you did something 10 years ago, have shelved it and gone back and thought, actually, now's the right time. I just wondered if you had any examples or any thoughts on that.
Well, yeah, we do that all the time. Sometimes we watch vicariously other people try to do something that we failed at, and we kind of realize what we might have learned by default. Sometimes some things advance and then we go back to them, so all of those. In fact, I was sitting in the audience listening to the previous talk and looked up what the most recent vouchers went for, and it's $150 million. So we've gone back from 80 to 150, and so my guess is that even that idea isn't, quote, a failure, it's probably a timing issue. So absolutely you want to do that. And sometimes technologies advance, sometimes pharma industry is flooded with targets, they don't really want any new ones, then 3 years later they don't have any targets because it's all they're getting barraged by Chinese assets, and all of a sudden they're saying, whoa, wait a minute, I got to invest in biology, otherwise I lose my differentiation. So there's lots of things. The problem, of course, is that if you don't have the resources to stay alive during that period, that's why, again, to me, as hard as it is to say, diversification kind of, for me, the kind of hard game, if you would give, sport that is entrepreneurship is about simultaneously surviving and thriving. Most people think that survival guarantees thriving. It does not. One thing we know is that without surviving, there's no thriving, but it takes different skills and different approaches and mindsets to thrive, but you first have to survive. And what happens is sometimes people say, I've arrived, I'm gonna just go after thriving, and they take their eye off of actually running out of money. So these are all things that, the good news, the bad news in what we do is that we live this times 45. So many, many more things are failing at the same time, but we're learning from them, hopefully, and go back to them.
Yeah, the time scale of learning is accelerating. One more question, and then I think it's time for drinks.
Was there a question here or there?
There was a question.
Oh.
Oh, okay.
Like that.
Please.
I was just wondering about what you were saying about the role for humans at the end of all of this. If AI is being trained on data which is based on observations and measurements that we currently know how to produce, do you see that there is a world that AI knows how to, or designs new ways of measuring things about the world, if that makes sense?
Absolutely makes sense. It's actually quite realistic and it's inevitable, no question. Because, you know, if you are able to create a model that has weaknesses in it because you're lacking data, then that model can tell you where you need more data. And if you don't have the instrument to generate that data, then you might go get that, you might go make that instrument. So I'll give you an example. It's probably not a perfect one, but just off the cuff of my head. So one of the areas we're interested in is making peptides or proteins out of all the amino acids. So when we went into that area, we thought, well, sure, we can get some models and do this. But it turns out there's no data whatsoever on how an all-D peptide would interact with an all-L protein, right? But now we know that. Well, actually, you need to be able to synthesize thousands of them to be able to get enough data to train a model. So then we had to go and invent a way to make an all-D synthesized peptide, which had some differences. So actually doing all of that with an AI kind of system, well, let's say a model, machine intelligence is what I like to call it because artificial seems artificial. So I think having that be an assistant in pointing to what the data lack is, either you go get it or you need to invent a way to get it. I think that's going to be a very interesting part of how the instrument business is going to evolve.
Okay, thank you. On that note, let's just thank all of our speakers in today's session.